In the world of biochemistry, proteins are the giants that drive life. But how do we define their scale? Welcome to a comprehensive guide on molecular weight. Molecular weight is the primary fingerprint used by scientists to identify, quantify, and characterize the diverse proteins that power every biological process. To understand these biological giants, we must first master the fundamental concepts of their mass. At its simplest, protein molecular weight is the sum of the relative atomic masses of every single atom within its chemical formula. The standard units are daltons or kilodaltons. 1 dalton is precisely defined as 1/12 the mass of a single carbon-12 atom. Proteins are built from amino acids, each contributing its own unique weight to the final structure. While weights vary from tiny glycine at 75 daltons to large tryptophan at over 200, the average residue weight is approximately 110 daltons. But weight isn't just a simple sum. When amino acids join via peptide bonds, a water molecule, roughly 18 daltons, is lost. To calculate theoretical weight from a sequence, we sum the residues and add 18 daltons back to account for the terminal hydrogen and hydroxyl groups. However, the primary sequence is rarely the whole story. Real-world proteins are often modified far beyond their initial synthesis. Post-translational modifications, or PTMs, can shift a protein's apparent molecular weight drastically, adding significant functional mass. Glycosylation can add several kilodaltons of carbohydrate chains, while even single-site phosphorylation adds 80 daltons, fundamentally altering the protein's profile. Complexity also arises from quaternary structure. Many proteins function as multimeric complexes, where multiple polypeptide subunits work as a single hollow protein. Measuring these biological giants requires precision. Let's explore the gold standards of the modern laboratory. SDS-PAGE is the lab workhorse. By denaturing proteins and coating them with a negative charge, we force them to migrate through a gel matrix based strictly on size. The distance migrated is inversely proportional to the logarithm of the molecular weight, allowing for routine estimation and purity checks. For absolute precision, we turn to mass spectrometry. Techniques like MALDI-TOF measure mass-to-charge ratios with atomic level resolution. This is the gold standard for identifying isotopes and small modifications, requiring only tiny sample amounts to achieve definitive results. To study proteins in their natural, non-denatured state, we use size exclusion chromatography. Here, larger molecules actually elute first. SEC is critical for determining apparent native weight and studying complex protein-protein interactions as they occur in nature. Choosing the right method depends on your needs, whether it's routine estimation with SDS-PAGE or absolute mass determination using light scattering. But why does this measurement matter? In biotechnology and medicine, molecular weight is more than just a number. In purification, it's our primary tool for verifying the success of protein isolation steps, ensuring we have the exact molecule we need. In the field of proteomics, molecular weight helps us identify thousands of different proteins within a single-cell lysate simultaneously. And in drug design, maintaining the structural integrity of biopharmaceuticals is paramount. Any shift in weight could signal a loss of efficacy. Molecular weight even serves as a diagnostic tool. Shifts in distribution are often early indicators of diseases like amyloid formation in the brain. From simple calculations to high-res mass spec, molecular weight remains the fundamental metric for characterizing the biological giants that define our health and biology.